Welcome everybody. My name's Cassie Sofing and I'd like to welcome you to our May 13th webinar. We're pretty excited about the content and what you all are bringing to our group. So thank you very much for joining us. If you have any questions during the webinar, please put them in the chat. There are 5 of us who will be monitoring it, and we'll welcome all your questions and we'll try to answer them in real time. What we'll be doing today is just a little overview. Dr. Rusty Lowe has an announcement. We have some mosquito stats, a mosquito notebook update, a sneak peek about our June webinar, and then our featured presentation. So hold on, it's going to be a wild and fun ride today. Many of you might know, but there's a citizen science conference coming up and actually going on. And Dr. Lowe.
So I just wanted to let you know that the work that you're doing as citizen scientists is truly valued and is real science, as you know. And I just wanted to call out 3 of our Globe Observer, Mosquito Habitat Mapper, and Land Cover citizen scientists, Mateo Kimura, Pratham Barbaria, and Prachi Ingle. They're actually all high school students, and they are presenting about some algorithms that they've developed to contribute to data quality for the Mosquito Habitat Mapper and Land Cover data. And so if you're not a member of the Citizen Science Association, we're actually going to, as soon as the conference is over, we are going to post their presentation in the blog. So you'll be able to take a look at it there. But here, just really quickly, you can just see some snapshots of these 3 amazing students. And they talk a little bit about what their plans are and how citizen science sort of fills in and what, working with the Globe Observer has done in terms of their career goals. They have 2 papers that are being presented.
All right, just a couple statistics for you to give you an idea of how 2021 is playing out so far. From January 1st until yesterday, we have 797 sites that have been observed with 2,197 observations. And of those sites, 168 include mosquito larvae photographs. So how are we doing since our last webinar? 311 of those are from our last meeting. 620 observations and 64 mosquito photos. Now I want you to remember this number because from this point moving forward, we have kind of a challenge ahead for you all, and we really want to see that number grow, so kind of keep that in mind. And my last kind of little bit of information for you all is that Citizen Science Con. It's a first of its kind online event series that invites everyone, everywhere to discover fun and important ways to help NASA do authentic research. There are sessions that will provide recipes to prepare for, participate in, and expand engagement in authentic NASA science. Imagine all the fun of a Comic-Con, only with fewer costumes and more science-y, save the planet, get everyone doing science kind of vibe, and you'll have NASA's inaugural CitSciCon. We hope you join us for some or all of the virtual event that will be in May 21st through May 22nd, because we want to celebrate you. NASA scientists collaborate with citizen scientists, the members of the public who volunteer their time for science, to conduct research they can't do alone. We have millions of citizen scientists who use their phone, their computer, or tablets to share local observations or analyze NASA data to help scientists discover galaxies, ground truth satellite data, track changes in weather and climate, and more. There's so much left to discover, so we hope you please join us. With that, I'm turning it over to Liz with the Mission Mosquito Science Notebook.
Hi, my name is Liz Burke, and I'm a member of the team working out of Alaska here. So one of my jobs is to work pretty exclusively at this point on the notebook that we are developing. And I know many of you are familiar with it. I've talked about it several times before, but it's an incremental study of mosquitoes leading up to building confidence enough that a student or a citizen scientist of any age could use the mosquito habitat mapper and move forward in their investigations and learning about that. So in order to get people at that comfort level and that confidence level, we're doing, as I said, these incremental steps. And this is our latest step. We fashioned this one after the hunters idea, the troll hunters and the monster hunters and that kind of thing. And so this is entitled Mosquito Larva Hunters. It's basically a standalone. We hope to be able to make this so people would not have to have done the entire notebook. They could actually grab this entry and do it as an independent study. So going with that theme of hunters, we have sent an official greetings note to everyone in which we're saying, oh, we are honored to have you join our team. And these are the things that we are going to expect you to help us with. And so your training is going to begin, and we want you to jump in now and start in this training with us. So we go on to the next page, and we just introduce, these are the three major genera that we focus on. So we use these as our models to show kind of what a larva is going to look like. And we list out the essential mosquito larva hunting skills that they would need. They need visual analysis and surveillance techniques. Again, saying with that same kind of a theme. And then we talk about building their visual analysis and surveillance, and that's where we start. So by starting with those, we go next to giving them some clues about how to do surveillance. And so within these clues, we're sort of setting it up to do an explanation based on evidence. So we're going back to, sort of some NGSS stuff in here too. But so we're giving you a clue. You're going to analyze that clue, and then you're going to be able to answer some questions based on just the information in the clue. So see how you can put your clues together and work forward from there. So we've listed 7 of them, and you can easily read them yourself. I don't want to take more time with that, but there are 7. And then you go on to the next page and apply those 7 clues to this lineup of specimens. And so we have 2 mosquito larvae in our lineup, and 3 of those are imposters. And it becomes the job of the trainee to go through and check off, oh, does this one have this trait? And then at the end, decide if it is really a mosquito or if it is an imposter. And so, and to make sure that you can get some feedback on that, we have also supplied the answer key to all of that. And so we have 2 other pages that give the answers to the questions that are in the line of the clues, and then the answers that go onto this table. And so that's our latest edition. It is now posted at this site that Cassie has posted there on the right-hand side, the globe.gov/web Mission Mosquito Review Resources, if you want to have a look at it. It ends, and I will end here with this. They get another urgent communique from the director of the Mosquito Larva Hunters group, and they are asked to assemble their toolkit because they are now going to be asked to go out in the field, put their knowledge to some practice, and see if they can track down some mosquitoes. So that's it. I hope you have a look at it. Thank you.
Thank you, Liz. Next up, I'd like to introduce Amelia Cook. And I had a chance to sit in on her session a couple months ago, and I was just like, oh my gosh, we need to share Amelia with everybody. She is an AP Environmental Science educator with the Norman, Oklahoma High School, and we've invited her to give a sneak peek of her work with the Choctaw Nation Oklahoma Jones Academy Virtual STEAM Summer Camp, where she uses citizen science and mosquitoes. She'll give her full presentation, Citizen Science for a Native Audience, at our June webinar.
Liz, that science notebook looks amazing. I'm actually going to be teaching with Jones Academy in person this summer for their STEAM camp, and that may be something that we start off with to kind of tie what we did last year to this. So that's a nice bridge there. That's a great work. But thank you, Chokma. Hello, thank you so much for inviting me to join you today. I'm Amelia Cook. I am Chickasaw. I teach high school science in Norman, and I am a PhD student at OU, kind of researching the tools of Native American storytelling to teach science. I'm looking forward to sharing with you how we use mosquito monitoring in a virtual STEAM, so science, technology, engineering, art, and math. And it was the camp for the Choctaw Nation's Jones Academy. And the students have an annual summer camp that was almost canceled last year due to COVID. The staff there was able to coordinate a virtual experience for their kids kind of last minute. And our team jumped in and we taught a full day. And we created a learning cycle that engaged our Native youth in citizen science, but also had this unique way of using their Native culture. We kind of really took advantage of that art in their STEAM camp. And it ended up being unexpectedly awesome. The collaboration was an incredible learning experience. It resulted in some very useful instructional strategies and some digital tools we used in this creative Native science curriculum, and ways to share Native ways of knowing and data that students were actually collecting themselves. The Globe Observer, the mosquito monitoring, really elevated our virtual learning environment and broke up Zoom fatigue. And students, we sent actual larvae. We didn't tell them the species, but we sent larvae to students at their homes, and they had cell phone microscopes, and they were able to, observe it. And we gave them a couple of larvae because, you want to poke at it and play with it. But they actually got to observe mosquito larvae and practice some of those identifications. And it was super cool. And the STEAM camp was really successful, and it was a positive experience for students and teachers alike. So I hope you're able to join us, I think June 10th. And I'll share more about how the buffalo bull and the cedar tree guided us to protect the circle of life using the science that we were learning. So look forward to seeing you soon.
Thank you very much. It'll be a treat to have you join us, so thank you very much.
Great. I'd like to introduce now our guest speaker for today, and that's Dr. Ryan Carney, a really impressive and cool guy in his own right. You can read a little bit about what he does here, but there are very few people that actually can talk about music and doing stuff with dinosaurs, and then also talking about mosquitoes. So he certainly is a really well-rounded kind of individual. And he has the wonderful distinction of being named as a National Geographic Explorer, which recognizes his uniquely gifted way that he inspires students and conservationists and scientists all around the world. So it's really, really great to have Ryan here. Ryan is a partner with our GLOBE Mission Mosquito campaign. And he's going to tell you today why we really need to amp up the amount of photographs that we are contributing over the next couple months to support the research that he is leading at the University of South Florida. So without further ado, Ryan, I'd like to just turn the floor over to you, and thank you for being here today.
Great. Thank you so much, Rusty, for that kind introduction. Much appreciated. I'm going to share my screen right now. So let me see.
Ew. Yes.
Okay. You, the mosquito larva. Should
Beautiful.
Be awesome.
Very cool.
That's a stock photo. No, I can't claim any credit for that one. But again, thank you so much for that kind introduction, Rusty. It's such a pleasure to be here talking with you all today. As Rusty mentioned, my name is Ryan, and I study two different things in my lab, dinosaurs and diseases. Quite disparate disciplines, but the technologies are all digital, and it quite often involves dead birds, albeit on vastly different time scales. So I study 150 million-year-old Archaeopteryx on one hand, and recently deceased birds on the other as sentinels for West Nile virus, which I'll talk about in a bit. Why study mosquito-borne diseases? Well, in addition to the fact that they are fascinating, it also has very profound public health implications. So when you ask people, what is the world's deadliest animal? Most people immediately say shark. They have their own week on TV, right? Shark Week. but they really kill only 10 people per year. When you compare that to everything else, including humans, it's really mosquitoes that are the deadliest animal in the world. And I should also mention that that number is vastly underestimated. So NASA estimates that it's closer to 2.7 million people die every year from mosquito-borne diseases. Most of this is due to malaria, but other diseases such as dengue. And that's just the mortality. You also have morbidity in non-lethal situations, which can also be devastating. So if you recall the previous global pandemic of Zika, just a particularly heartbreaking disease given the severe birth defects, things like microcephaly, daily seizures, and things like that. So, robbing a child of their entire livelihood, their full potential before they're even born. So just even when there's no mortality, just the morbidity associated with these mosquito-borne diseases can be very life-shattering. So tragically, there are no cures and very limited vaccines for most of these diseases. And so really the primary way to fight them is through mosquito surveillance and control. So figuring out where those diseases are and specifically where those mosquito species are. And the key here is that it's been estimated only about 1% or less than 1% of mosquito species actually carry and transmit disease to humans. So being able to determine where those specific mosquito species are in the world at any given time, also to be on alert for invasive species that may be carrying new diseases or that could outcompete local mosquito species is critically important. And that's one of the themes that will be carried forward throughout this talk, is being able to identify species in real time and really using automated AI scalable solutions to do so. Because the other key part here is that identification, whether it's mosquito adults or mosquito larvae, is very time intensive and very resource intensive. And so if you can use a smartphone and artificial intelligence to scale that up in real time and then map it and model those habitats and diseases, then that's sort of our overarching goal here. And so the outline for the talk is basically going to encapsulate 3 different main points. I should also mention that this is part of a larger 4-year NSF project. So right now we're just in the beginning phases. So this is the 7th month of this 4 years. First, I'll be starting off with a little bit about geographic information systems, or GIS, specifically the DYCAST algorithm that's useful for looking at clustering phenomenon, whether it's West Nile virus or dengue or Zika. And then also habitat modeling, specifically for mosquitoes like Aedes aegypti. Secondly, I'll be showing some of our preliminary results from the artificial intelligence work that we've been doing, specifically on mosquito adults, as well as our future plans for mosquito larvae, which is where the mosquito habitat mapper comes into play. And then throughout this, I'll be talking about different ways that you can become involved, whether it's with the mosquito habitat mapper or some of the other related apps and tools as well. So as I mentioned, that includes Mosquito Habitat Mapper for mosquito larvae. And there's 2 other apps that I'll be talking about throughout this, a Mosquito Alert and iNaturalist for basically taking photos of adult mosquitoes. All of these photos are useful in training our artificial intelligence algorithms so that in subsequent years, we'll be able to have a real-time automated identification system where we'll be able to get real-time feedback that, yes, this is Anopheles stevensii that you just took a photo of. And also, before I begin, I want to give a shout out to all the wonderful collaborators on this project. So everyone on this slide, as well as others, such as John Palmer from the Mosquito Alert team and all of his collaborators, Darrell Capan at iNaturalist and that team as well, Larry Reeves and Nathan Burkett-Cadena at the Florida Medical Entomology Laboratory, and of course the many, many mosquito control districts that we have partnered with. So all of the work that I'll be showing today is very much a team effort involving a very big team indeed. So thank you to all the individuals and institutions here. So as far as my own background in mosquito-borne diseases, so I'd always been fascinated by diseases, especially zoonotic and vector-borne diseases. So I remember reading The Hot Zone when I was a kid and being fascinated by Ebola. My first academic position, I shouldn't really call it a position, it was a job. My first academic job was as an undergraduate student in a Lyme disease lab where it was my job to collect tick saliva using micropipettes. So I would extract the saliva out for a blood complement study that Dr. Lane was doing. That then set me up to get a position at the California Department of Public Health right at the time that West Nile virus entered the state. So West Nile virus was first found in Southern California in 2004. Just a little bit of a quick background on West Nile virus. It's a flavivirus, so it's related to things like dengue and ZIKA. What's different about West Nile virus is that birds are the natural host. So there's this amplification cycle that goes through birds, and it's only incidentally that the disease is transmitted to humans. That also means that birds can be very sensitive surveillance indicators for the spread of the disease. Now, West Nile virus was first detected in the United States in 1999. To date, there have been over 3 million infections and approximately 2,400 deaths. And again, I began at the California Department of Public Health in 2004. And in 2005, it was really the epicenter of West Nile virus in the world. It was 880 human cases with 19 deaths. And my job at that time, my position was coordinator of the West Nile virus dead bird surveillance program. That's the dead birds. And so it was my job to get all of this information from throughout the state. We had a hotline of a dozen people taking calls from residents who, there were crows literally falling out of the sky that we would initially test, but then we would report in this GIS database and send that data to the local mosquito control districts for their surveillance and control. So a lot of information, and I really wanted to leverage that data and see if there was some way that we could not just report where those dead birds were, but really use that to forecast risk to the human population. And so I set up a collaboration with Sean Ahern and Al McConkey, who are at the City University of New York, who had developed an algorithm called DYCAST, which is Dynamic Continuous Area Space-Time System. Now, the way that it works, it detects clustering of dead bird reports in space and time using 5 biological parameters of the virus, the vector, and the host. It predicts areas with elevated risk to humans, so you can construct things like daily risk maps and animations. And in 2005, we started a pilot project in California, specifically in this area of Northern California, the Sacramento and Yolo counties, because it was predicted that even though West Nile virus hadn't shown up there, that's where it would be kind of the hot spot based on, so certain environmental factors and all of that. So it was thought that would be it. So we set up a pilot project. The way that the algorithm works, before I show you some of the results, you take your area of interest, you overlay a grid. This is all done in GIS software. That grid is composed of different tiles or cells, and the DYCAST analysis is run on each one of those tiles. Now, the spatial domain there, that circle, is based on the flight range of the particular vector. So for West Nile virus, we're looking at the flight range of Culex genus mosquitoes, the maximum flight range, and then we would map the dead bird reports, which are shown here as asterisks. So that's in space. We can stretch that vertically in time and look at those dead bird reports in space and time. And that time dimension is based on the incubation period within the bird hosts, as well as the extrinsic incubation period within the mosquito vector itself. So here we look at those asterisks, those dead bird reports, and look for clustering in space and time. So we count the number of close pairs, and we do a Knox test and Monte Carlo simulations to see if there is statistical significance. If there is, that means that it's high risk, and we light that tile red. If there's no risk or low risk, we leave that empty. So it looks a little bit like this: unlit tile and a lit tile. And we can then map on the human cases. So here they're shown as dots, and we can see predicted humans and missed human cases, depending on if they were in that same tile before or after it was designated as high risk. So some of the initial results and what's in the paper cited here are shown here. And I'll show you the epidemic curve in the next slide, but just to show what some of the daily risk maps and animations look like. So you can see those incipient clusters there, those 2 red blobs. Start to see some of the human cases come in there as well. Spread it out. So again, this was the largest West Nile virus epidemic to date. So showing that what that looks like on a timeline here, the human cases are in black, and red shows the DYCAST risk denoted by the number of high-risk tiles. And so at the very end of our pilot project, this is the results, those two red blobs. So we didn't know that the human cases were there, those two black dots. We just had the red, and I immediately sent those results to the district manager at the time and said, there are these two areas of risk that is, predicted by this model. They immediately went and put mosquito traps in the exact center of those two clusters, and within 48 hours had the first West Nile virus positive mosquito pools in Northern California. Now, we didn't know at the time, but when we got the human case data and mapped that on, we saw that these two clusters also predicted the first two human cases in Northern California. So very promising initial results, and we were able to get full funding for the rest of the year to do this for the entire state. And it was then adopted as part of the permanent state surveillance and response plan for West Nile virus. We did things like create a GIS dashboard. This was led by Chris Barker at UC Davis, where we were basically hosting the DYCAST risk that you can see here, along with the different surveillance elements, so mosquito pools, positive sentinel chickens, as well as some of the other test results. So it was kind of this one-stop shop that the local mosquito control districts could log into, look at the surveillance in their area, and have an informed decision as far as where to do additional surveillance and control. So this shows some of the results for the rest of the year. At the far right there, that shows a heat map as far as the number of days that tiles were lit. So the deepest red is about 100 days there. And so one of the keys is that early detection is key. So at that beginning of the epidemic, that's really before things really kind of caught on fire and exploded throughout the region. And so if you can use things like DYCAST to find those initial clusters of transmission, you can go in and eradicate the disease, eradicate the mosquitoes before a larger epidemic occurs. Also because, I should also mention, overall, 93% of cases within Sacramento County were predicted by the model. This was 82% for the entire state of California. And this was all 37 days prior to the onset of illness. So plenty of time to do additional surveillance as well as control measures, which I'll talk about very briefly shortly. And because I like things in 3D, I just wanted to show the risk in 3D. So if we rotate this, and now on the x-axis is time, we can see the beginning of the epidemic on the far left there, which is actually the city of Davis there. As kind of the epilogue to that story, the DYCAST results, as well as some of the others that you saw in that GIS dashboard, were actually used to direct surveillance and control, specifically this aerial spraying of mosquito adulticide to kill the infected mosquitoes during this emergency situation, which again was the largest epidemic of West Nile virus to date. And this study really came about because I was looking at the DYCAST risk animations and saw some of the risk fall away and dissipate. And so I asked the district manager, why do you think this might be? Do you have any anecdotes? And he said, well, actually, that's exactly where we did spraying. And so I said, well, let's take a look at that. So we got the GPS traceways of the aircraft that were flying over the region. And then we're able to find, by plotting the human cases onto the GIS map, that there was a statistically significant reduction in the number of human cases in the sprayed areas compared to those areas that were not sprayed. And if you account for the West Nile virus incubation period, which has a maximum of about 14 days, there are actually no new cases in the treated areas, whereas there were plenty in the untreated areas, and the odds of infection there were 6 times higher. And so really the punchline here is that these risk maps were used to target mosquito control, surveillance, and public education campaigns, and it really demonstrates the utility of using GIS for prediction, as well as response and evaluation. That's what we're trying to carry forward. So later on for my master's work, I had adapted the parameters of the DYCAST model for dengue. Now, dengue is much more severe of a flavivirus. It has something called antibody-dependent enhancement. So if you get it more than once, then you get really bad outcomes, or you can get very bad outcomes like dengue hemorrhagic fever, dengue shock syndrome, things like that. There are 50 to 100 million infections per year, and over 2.5 billion people, 40% of the world's population, is at risk for dengue. So quite an important disease. And what you'll notice here with the parameters, starting with the flight range, it's much smaller. So the principal vector of dengue is AEDES aegypti, so different mosquito, the much smaller flight range. There's also no birds in the amplification cycle. And so it's just the human incubation period as well as a longer extrinsic incubation period. But the model itself worked the same, although on a different software platform, which we'll talk about shortly. The results look like this. So you can see the epidemic at the bottom left. That's the timeline with the number of cases in black. There were about 5,000 confirmed cases and about 10,000 unconfirmed cases. And what I want you to pay attention to on the map there, so the human cases are shown in black, but in that area that's encircled in blue, you'll see just a little blip of risk. But otherwise, if you were to just look at those cases, you wouldn't see a cluster. So I'm just going to start playing the video. So you can see that little blip of risk that the algorithm detects as statistically significant, but just looking at that, you don't really see any risk. So that was during the winter. Now, as the temperatures heat up, the disease really amplifies and catches fire in that same area. So that exact same spot that was initially detected once winter hit was exactly where it reemerged once the temperatures increased, and you got this huge conflagration over the entire area. And so that suggests that the model detected what's called the cryptic interepidemic focus of transmission, which is just a complicated way of saying that that's where the virus was kind of hiding out during the wintering months. And it's really the holy grail for mosquito control, because if you can target those areas, if you know where the disease is hiding out, you can really seek and destroy that before it, it reemerges come springtime and summer and really lights the whole rest of the city on fire. And so that was one promising finding from this. I should mention this was run retrospectively as opposed to in real time and prospectively, like in the California West Nile virus model. But the model predicted 90% of cases and 66 days prior to onset of illness. So again, we're looking at clusters of human cases to predict future human cases. The West Nile virus model was clusters of dead bird reports to predict future human cases. More recently, we've looked at epidemics of dengue and ZIKA in a nearby town of Campinas, Brazil. So I'm just going to show the animation here. We have dengue in red and orange and ZIKA in black and gray there. So two concurrent epidemics that have almost identical parameters as far as the model is concerned. It's the exact same vector, so AEDES aegypti. So we're looking at the spatial distribution of the ZIKA hotspots versus the dengue hotspots. But another thing that we're doing, and which is part of the grant here, is we want to develop this into a tool that anyone around the world can use for free. So we are creating a version of the DYCAST algorithm that runs on open source software, QGIS, which is open source GIS platform, as well as PostgreSQL. So if you have, for example, dead bird reports in a PostgreSQL database or human cases of dengue that you want to analyze, you'll be able to download our plugin once it is finished and released. And within the QGIS environment, pull the plugin up, as you can see here, boxed in red. You'll enter in the cases, the different parameters as far as the spatial domain, temporal domain, things like that, depending on what disease you're looking at. And then you'll be able to generate both the static daily risk maps as well as create animations such as this, which is the data from Campinas. So what we would like to hear from you is, do you use QGIS, and what sort of features might you like to see in such a plugin? So again, we're developing this over about the next year, but we'll be maintaining it through at least the next 4. But what sort of features would you like? Would you use this for any of your West Nile virus deadbird surveillance, for anything like dengue or ZIKA, or perhaps other clustering phenomenon as well? Also, if you are interested in using this and are a Mac user or are Python savvy, we'd like to hear from you as well, because we want to do a lot of beta testing on that platform. So it will run on Mac and PC. Again, all of this is free and open source. You can go to our temporary project page at diecast.org, download any of the previous papers. There's a couple links to the GitHub pages where you can download the previous versions of the software. And that's also where we'll be hosting the finalized version once it is fully developed. Also, just to talk a little bit about more about AEDES aegypti with respect to modeling its habitat. So this is not DYCAST related. This is separate analyses within the GIS environment. This is also, I should mention, both this study and the next are student projects. So this is a master's student in public health department. And here she looked at AEDES aegypti habitats in Barcelona, Spain. And I'm going to show the animation here. This is in arc scene, which is why it's shown in 3D there. And looked at basically narrow streets, high population density, urban vegetation, and courtyards to look for high risk of Aedes aegypti populations to use as a risk map. What was not used was any data from mosquito traps or any validation data. That was something that we did subsequently a little closer home in Florida. Now, I should mention that these results I'm going to show are hot off the presses. This is from honors student Connor Mapes' thesis that he turned in literally 2 days ago. So just starting off here with some background, this is the Tampa Bay area, population of about 3 million people. And so here, the local mosquito control districts in these 4 counties, Hillsborough, Pasco, Pinellas, and Polk, were kind enough to provide us with trap data over many, many years. Here we analyzed 2017 to 2019. We also got high resolution aerial photography from the Southwest Florida Water Management District. And when I say high resolution, it was 0.5 meter resolution. So we actually had to do some down sampling for some of these calculations. But this is the study area and some of the trap data. And Connor then generated various maps. So the first 2 in A and B are MaxEnt, maximum entropy, ecological species niche model. And so the way that this worked is he found the present data of Aedes aegypti, so whether it was present at a trap location or it was absent, and then was able to find correlations with vegetation for A and different types of land use and land cover in B. So in B there, he looked at things like, all right, was the trap in woodland or wetland commercial area? And what he found, one of the key findings, was that it was the recreational land use classification that was the highest, had the highest association with Aedes aegypti populations across all 4 counties and in both seasons. So he ran this set of models for both winter and summer. And so it was really recreational all across the board. In C there, that shows the abundance data. So while A and B showed the presence absence of Aedes aegypti, C shows the abundance of Aedes aegypti found in the CBC traps. And the IDW is just inverse distance weighted. It's a type of interpolation to calculate what an estimation of the abundance would be between those areas that were actually sampled. So essentially an abundance map there. And then D shows the population density in those 4 counties, the human population density. So from that, 2 different maps were created. This just shows that one of the starting data sets, that abundance data. And you can see the geographic scale there. The scale bar at the bottom is 20 miles. A map like this isn't very useful for mosquito control districts because, yes, they can see what traps had high abundance, but it's not operational in the sense that they can take that map and say, okay, well, we need to go to this park and put a new mosquito trap there, or do other types of surveillance and control. It's not that fine scale enough. And so what Connor did was to take this abundance, multiply it by the 2 MaxEnt models. So remember the association with the vegetation and the land use to create a high resolution, fine scale vector habitat index map on the left. So this shows within one of the counties, Pinellas, very, very high resolution map, a heat map, where the areas that are red show those that are most likely to have high abundance of Aedes aegypti. On the right, this is the vector habitat index multiplied by the human population density as a proxy of risk. So you have vectors on the left, and on the right, it's vectors times the hosts. So 2 different types of maps that can be used operationally by the local mosquito control districts to direct further surveillance and control efforts. So as far as some next steps, Connor over the summer will be creating a GIS dashboard to host some of the habitat models, like what was just shown in Florida, as well as some in Ethiopia, as well as to map the results from the citizen science campaigns from Mosquito Habitat Mapper, from Mosquito Alert, and from iNaturalist. So it will be this one-stop shop where you'll be able to go, you'll be able to see the citizen science results, as well as the habitat maps. And so the thinking is that the fine scale habitat maps will help direct citizen science as well as surveillance and control by the control districts. And conversely, the citizen science data should provide very valuable validation data for those models. I'm just going to quickly go over this. This was kind of a fun prototype that we did with remote sensing data. You can extract out different biosignatures for breeding habitats. We all know mosquitoes breed in water. One of the keys for Aedes aegypti, for example, are abandoned tires. And so we created a biosignature of a tire rubber. And we were able to use satellite imagery and process it in a way that we were able to detect abandoned tires in Hillsborough County. And then sent students out to validate those and had well over 90% success rate. And so they looked a little bit like this, where sometimes it was a pile of tires, sometimes it was an isolated tire on the side of the road. But the results helped us locate these tires. But then we also want to say, okay, well, is this something that we could then show on a cell phone? And we do something similar to Pokemon Go, where it can be geo-referenced and using a 3D model to basically pinpoint users to that location. And so we created this fun prototype. It's not something that we will be doing or should be, but rather something that we could do and wanted to check out its potential utility. We nicknamed it Mosquito Go. Basically, it works by having the GPS coordinates of your points of interest, such as tires that are detected from satellite imagery or drones. And then you just kind of zoom in as if it were a Google Maps. You can see 3 around there. And then you'd switch to the augmented reality mode. That's what the AR stands for. And then you'd see the 3D floating tire there, which directs you that there is a abandoned tire outside. So then the student walks outside. This takes a few seconds. So I'm just going to kind of jump through here and follows the tire up to the tree where there is an abandoned tire, which of course we put there as well as the GPS coordinate, but it shows that we can geo-reference some of these augmented reality 3D models to use as whether it's a gamified app or just a fun project as it was here. Okay, so now from AR to AI, I'm going to show some of the initial artificial intelligence results. So this is from a paper last year. On the left, you'll see this video of a student taking a photo of an adult Anopheles crucians. Setting that out, and then he's going to open up the phone, zoom in with the camera, and take a picture. That picture is then sent locally to a server on campus, and it will do its calculations, which take a second. While that is running, I'm just going to explain a little bit about what's going on in the background. At this point, we had already trained the artificial intelligence algorithm to detect various species using thousands of different photographs. What the algorithm is doing right now, this is based on a convolutional neural network, or CNN. It's creating a class activation map, or a CAM, which is a heat map that shows the pixels that are used in the classification by the AI. And so shortly, you'll see that result pop up on the app. And indeed, it was Anopheles crucians, 98% probability, and there's the class activation map showing the pixels that were used in that classification. What's also interesting from these results, if you look at the classification accuracies here, that shows that the 9 species that we looked at separated into the 3 critical genera, so Anopheles, malaria vector, AEDES, dengue, and ZIKA, Culex, things like West Nile virus, for example. And if you look at the diagonals, which is the key there, that shows the classification accuracies. So for the Anopheles stevensi, for example, that first tile there is 0.93, so 93% of them were correctly predicted by the algorithm. And what's interesting here is when you sort of couple the artificial intelligence with natural selection. And by that, I mean, what you see there, that tree is what's called a phylogeny, or an evolutionary tree that shows the relationships among the various taxa. So for example, in the Anopheles, you have Anopheles stevensi diverge evolutionarily first, and then the lineage from that diverged into the what's called sister groups of the Anopheles crucians and Anopheles quadrivividatus. And so what you see here is that the highest classification accuracy was actually for Anopheles stevensii, which is one that diverged first. So it had its own evolutionary trajectory, which went further back in time. So the further you go to the left, the further back in time, and that black dot, there's the common ancestor for all of those mosquitoes, which is about 217 million years ago. And so that longer branch length, that longer evolutionary time for Anopheles stevensii, we would assume would equate to some anatomical differences, right? That there'd be some morphological differences of stevensii compared to crucians and quadrimaculatus. And so it's interesting that that was the species that we found had the highest classification accuracy. We found the exact same pattern in the AEDES clade, where AEDES aegypti was the first to diverge within that group, and it had the highest classification accuracy by over twofold compared to the Informatus and Tanuricus. So very initial results, but again, really interesting as far as the interplay between the artificial intelligence and the natural selection. So moving on to the next architecture that we utilized, this is the mass region-based convolutional neural network. And most simply, it works like this. So you have a photo of, in this case, obviously an adult mosquito, do a box-wise segmentation, just followed up by a pixel-wise segmentation or masking for each of the anatomical regions. So we call this anatomically informed deep learning. So the pixels are extracted out for each anatomical region, and the algorithm is then run on each one of those independently. So the deep neural network may say, okay, for this thorax, it detects a white liar pattern, which is indicative of AEDES aegypti. And so after running through the logistic regression classifier, it will then identify the genus and species as AEDES and aegypti. That's sort of how it works again, anatomically based deep learning. And so the results look something like this, where the different regions of the anatomy are color-coded, and they each have their own probabilities. So you can see what the relative importance of each anatomical region is in the case of AEDES aegypti for that particular specimen, or Anopheles crucians. And because it learned to recognize wings and legs and thorax and things like that, it's also useful for other insects. And so we have some work ongoing looking at things like bees and bee mimics. Now, the utility of that, again, is really to detect those few species, 1% of species that are medically important, that are transmitting diseases to humans, but also any invasive mosquito species that are new to a region or that may be harboring new diseases. And by combining the power of AI with citizen science, we can really leverage these technologies and approaches to create a global surveillance system. And so there's some really just wonderful success story by our colleagues at the Mosquito Alert in 2019, where their app, Mosquito Alert, provided the first detection of Aedes japonicus in Spain. So this was found by a citizen in the area, took a picture, and then there was some follow-up for confirmation to show that it was indeed Aedes japonicus. And so again, our hope is by coupling citizen science infrastructure with things like artificial intelligence, we can have this real-time, automated and scalable approach for identifying those species that may be transmitting disease or that are invasives. And so I encourage you all to check out Mosquito Alert. There's also a link through our project site, which has the URL of mosquitoai.org. So you can check that out there. So this is what we encourage you to use for mosquito adults, as well as iNaturalist. And very recently, we launched an iNaturalist campaign specifically looking for certain invasive mosquitoes in the Caribbean region, as well as Central and South America. So I'll talk a little bit about that as well. But these two apps are great for sending pictures of the mosquito adults. And as we talked about before, the key right now is to get as many photos as possible, especially of these target species, so that we can use those photos to train the artificial intelligence algorithms so that in the future, we'll be able to detect them using the AI in near real time and ultimately real time. So you'll be able to take a picture of something and get immediate feedback that, oh, this could be an invasive species, or this is something that could be spreading this new pathogen in your neighborhood. And as of this morning, so this is a recent screenshot here, over 400 observers to our campaign. We'll also be having a BioBlitz this summer as well, which I'll talk about shortly. So we launched the campaign last week. And our posters actually got picked up by the local news when they went to interview our colleagues at Polk County. So I'm going to show that right now.
Polk County is on the lookout for a new mosquito species scientists say carries yellow fever. Dr. May News 9's Ashanti Ford spoke with experts who are working to make sure it doesn't invade the area.
Mosquitoes. You just started itching, right? Well, this flyer has been circulating around Polk County showing a new mosquito species that's headed for our county. And even though we can make light of it with the flyer, I'm told this is not a bite you want to receive.
I spend a lot of my time on surveillance and data monitoring and doing a lot of ID.
Jackson Mosley is an entomologist for Polk County's Mosquito Control Unit, which basically means if you have a question about an insect, this guy has the answer.
We were talking about the CDC trap, right?
A series of tests are run on mosquitoes so that Mosley and his team know how to manage them. So when this new species popped up, scientists found that Florida met this bug in the early 1900s.
It was thought it was an isolated case, but now they've found some more that have moved past the Keys.
And Polk County, I'm told, has the perfect habitat for this mosquito that scientists over at the University of Florida say is carrying yellow fever.
Yellow fever was something that early on was a major player in disease in Florida.
But the good news is there are no signs that it's here now. And be assured, Polk County's mosquito control team is already on the lookout with dozens of mosquito trap sites around the county.
The routine to check all 60 plus sites.
Mosley leaving us with a tip.
Wear products, repellent that carry DEET, and make sure if you have to go out after, dusk, to wear long sleeves.
I'm Ashanti Ford with Spectrum Bay News 9.
Okay, so basically, Jackson was talking about Aedes scapularis and why, you can't hear me now, right? All good? Cool.
Yes.
Okay, so Jackson was talking about Aedes scapularis. This is important because it transmits 15 different viruses. So this includes yellow fever and St. Louis encephalitis. And its last known whereabouts, as you may have at least seen in that clip, was Miami-Dade and Broward counties. So what's interesting about Aedes scapularis is that it's a floodwater mosquito, meaning that it will lay its eggs. They are desiccation resistant, so they can dry out. And then when the floods come or when water touches them, then they all emerge. And they can emerge in numbers that are actually abundant enough and dense enough to kill cattle. It can be that bad. Now, not only do they lay their eggs in areas that could get flooded, they also have great ecological plasticity and will lay their eggs in artificial containers, which is one of the big dangers here because they have adapted so well to man-made environments. So previously they were known from South America and Central America. And more recently, and this is a paper published by Larry Reeves and colleagues in 2021, just this year, some spots in southern Florida, Miami-Dade, and Broward counties, specifically where Aedes scapularis is found. And so what we need right now is as many photos as possible of Aedes scapularis with which to train the artificial intelligence algorithms so that we can detect it in real time across all of the platforms. Mosquito habitat mapper for the scapularis larvae and mosquito alert and iNaturalist for the adults. All right, so not just in Florida, any of those areas that are shown in green there, we need photos, and especially of the larvae. So we don't have any photos of the larvae. We have a few of the adults, but really we just need as many photos as possible. The more images we throw at the artificial intelligence, the smarter it gets. So that's Aedes scapularis. Next up is Aedes vittatus. So you can see there's 3 pairs of spots on the sputum there. So it's fairly salient as far as the markings. This transmits 7 different viruses, including ZIKA, dengue, West Nile virus, and yellow fever. Its last known whereabouts are Cuba and the Dominican Republic. And so if you are in any of those areas, please send us as many photos as possible of Aedes vittatus or anything that you think could be Aedes vittatus. And there's a paper, again, from just this year, talking about how it has now been introduced into Cuba and the Dominican Republic. So we're trying to detect this invasive. So the third invasive that we're looking for is something that we don't actually have any good photos of. So this is Aedes MEDIOVITTATUS. It's the Caribbean treehole mosquito. It's a vector of dengue. It's found in forested and rural areas, but it can also lay its eggs in both natural and artificial containers, including bamboo. So it's found in these regions. If you go to something like gbif.org, there's actually very few specimens have actually been collected and are found in museums. But Arizona State University was kind enough to send us photos just a couple days ago from theirs. This is courtesy of Andrew Johnston. This is one of the Aedes MEDIOVITTATUS specimens that they captured in Puerto Rico a number of years ago. And so we plan to launch a BioBlitz on iNaturalist this summer for any of the regions in the Caribbean, especially Puerto Rico and the Dominican Republic, because there's actually zero observations for this species on iNaturalist. So if you are in any of these areas, please be on the lookout for this mosquito that may be in tree holes, maybe in artificial containers, but it's Aedes MEDIOVITTATUS, and we need the photos to train the AI. The last species that we're looking for a little bit further away is Anopheles stevensii. So when you typically think of a malaria vector, you think of Anopheles gambii, which is more of a rural mosquito. Anopheles stevensii is different because it will lay its eggs in artificial containers, sometimes even in the same ones that Aedes aegypti may as well. And so the real danger here is that it has been recently introduced into Africa, specifically Ethiopia, which is where we're having our campaign right now. And the real fear is that it's going to ignite these urban outbreaks of malaria because it is this mosquito that likes to lay its eggs in artificial containers, unlike Anopheles gambii. And so we're trying to develop this AI as fast as possible to detect both the larvae and adults of stevensii so that we can use citizen science and use the AI to detect stevensii when a user uploads a photo, and also do habitat modeling so we can find areas that it may already be or that could be in the future. And so we're trying to leverage technology to really combat this, this deadly invasive vector. And here are two recent papers from last year on Anopheles stevensii in Africa and Ethiopia. So what can you do? Take as many photos as you can and upload them through Mosquito Habitat Mapper. Everyone listening is very familiar with this tool, of course. And there's just wonderful resources online that will show you how to go through all the different steps. One of the images that I really liked was this one showing the larval anatomy. And this also underscores one of our other next steps for the artificial intelligence is that we need to train the algorithm to detect, all right, what is the head versus the thorax versus the siphon, et cetera, et cetera. And so what we need to do is mask out the different portions of the anatomy, train the algorithm, which looks a little bit like this. And so we're looking for volunteers to help us in doing this masking step so that we can train the algorithms and then ultimately deploy it. So if you're interested in helping us with the masking of the mosquito larvae, please visit our site at mosquitoai.org. At the very top, there's a note if you're interested in participating in this. Please visit our registration page here. That'll bring you to a registration page where you can enter in your info and email. And then we can contact you with further information and images once we get all this started. Now, as far as taking the photos themselves, there are sort of three things that we need, again, for the training of the artificial intelligence. So first, we need a full body image that shows all of the hairs. So there has to be enough water that the hairs are kind of floating out to the side and not against the body. So the full body with all the hairs. Second, we need at least 1 to 2 photos of the tail. So segment 8 there, along with the siphon and the saddle and all of the hairs and brush that you see there. And third, we need the head and thorax with all the hairs. So again, we're trying to teach the AI to recognize the different anatomical regions, and the hairs can also be very diagnostic as well. So we need to have those hairs present and also in focus. And so just some tips here. And wrapping up, I want to really convey the message that in-focus images are really key. We need to have each photo in focus and as much of the mosquito larva in focus as possible. And so many of you have used the 60x clip-on lenses before, so they have a light on them. You want to turn the light on, the white light, not the UV light. There's also a clear collar, which will be flush with the table when you have the larva underneath it. There's also the 1000X, which is great for species level ID. They're under $20 on Amazon, so those are great as well. But the 60X is definitely sufficient, especially since those are the types of photos that we'll be analyzing through the Mosquito Habitat Mapper data once the AI is up and running. So some tips here. First of all, when you have the clip on there and you zoom in, by stretching the screen or something like that to zoom into that larva, you can then tap the larva so that the phone will focus in on that. So the camera will know, okay, you tap there, I'm going to focus in on that. And so that can help focus on the larva, number one. Number two, we found that it's helpful to turn the HDR off. This is especially important if the larva is moving. And if the larva is moving, you can always euthanize it with ethanol or also cool it down. Just stick it in the refrigerator for a while, and that will reduce that movement and give you a clear shot and more in focus image. Fourth, depending on your phone and depending on the case, it may be prudent to take the case off. So again, it really depends on the phone and the case, but sometimes that can affect the focus. We've also found, so this is just a recent discovery, is that using a washer, so basically putting a washer on the table to elevate that clear collar of the lens up by about 1 millimeter can actually help the focus. And so if the larva, for example, is kind of floating above the meniscus and it's kind of elevated, having that washer there will, in some cases and in some phones, provide for better focus. And so that's something to keep in mind. Just a 3/8 inch washer can do the trick, but that's something that allows you to elevate it off the table just a little bit and in a stable fashion. And the last two, which are less tips and more just requests, we need larval absence data. So it's good to have images of the larva, but we also want to know where they are not, which is important for the habitat modeling. And to that point, we also really need land cover data. So whenever you finish doing a mosquito observation, it would be great if you could then go to the land cover tool and then take an observation there so that we have that environmental data to couple with the presence or absence data of the larva. And so that will really help our mapping and modeling analyses down the road. So just to wrap up here and summarize, Mosquito Habitat Mapper and land cover. On the AI training for the masking that I talked about, if you're interested in volunteering, contact us through mosquitoai.org. For the ID, so if you know of any, for example, local entomologists who are trained in larval mosquito identification, please have them contact us because we need to essentially calibrate the system. We need to teach the AI that, okay, for this genus of larva, it looks like this. And so we need to go through a lot of the data that's already in the database that has been collected and specify what is the genus associated with that. So that again helps in our training. And lastly, if you or someone you know has experience in GIS, especially spatial statistics or Python programming, and is interested in joining our team as a master's student or a postdoctoral researcher, please contact us as well. And so we have 2-year positions available for each of those. And so the fourth kind of goal in this workflow here is to train and then ultimately deploy the AI so that we have real-time feedback to the user, but also we can map any invasive. Once it's detected, we can notify the user that this is a new species or you have one that is of medical importance, and help establish this, as I mentioned before, a global surveillance system that is real-time and leverages both the citizen science platforms of Mosquito Habitat Mapper, Mosquito Alert, and iNaturalist, along with the computational artificial intelligence, DYCAST cluster modeling, and habitat modeling as well. And so we'll be able to scale up the identification and find those 1% of mosquitoes that are transmitting diseases to humans, as well as detect any of those invasives. So we can create things like real-time maps, whether it's disease risk maps or distribution maps, that mosquito control can then use for targeting, surveillance, and control, and ultimately lead to fewer mosquitoes and less disease. And so with that, I'd just like to show one last video, and please feel free to reach out to us at any time at mosquitoai.org, and I'm happy to answer any questions. Thank you.
It's great, Ryan. Really, truly amazing. We do have a few questions, and while your video is playing, I'm going to read you the first one.
Sure.
Is there any information on the sudden death mosquito and its effects of Aedes aegypti populations?
You say that one more time, the sudden death?
It's on the sudden death mosquito and its effects of Aedes aegypti populations.
I'm not aware of what that mosquito would be.
I can sort of address that. What they're talking about, Ryan, is Oxitec had developed something they called the sudden death mosquito back in 2008.
Gotcha.
I don't think they're using that terminology anymore for this. For the most recent genetically modified release of mosquitoes that's taken place in the U.S. is one where they mate and don't have a live progeny. So I don't think the southern death mosquito, as far as I know, and I could be wrong because this is not my field, but I haven't heard about that for a number of years. And I think they've moved on to a different type of engineered organism.
So what would be interesting is if they differ at all morphologically, whether the fact that they were reared in a lab or just if there is some sort of phenotypic consequence of the genetic modification, if there is anything that is different morphologically, the AI might be able to actually detect that. So what we would need is a sample of those Oxitec mosquitoes and just take a whole bunch of photos of them. That's something we've thought about before, but is not something that we've necessarily taken any steps to look at, but definitely something that's been in the back of our minds because, yeah, that would be cool to check that out if there are any anatomical differences. They may not, or it may be, that lyre shaped pattern may be so, distinguishable, and that may be all that the AI hones in on. So who knows? It is theoretically possible that it could distinguish that, but we won't know until we try.
Thank you. There are a couple other questions, but I just wanted to quickly wrap up what we have. Is the reason why you're here is to encourage us to get mosquito larvae observations. And so we've got just the perfect opportunity to ramp up our photographs July 25th through August 25th. We've got a land cover mosquito habitat challenge coming up, and more information will be on observer.globe.gov coming up. And then our next webinar with Amelia Cook and on citizen science with native audiences. So if I could get everybody to do two things, take mosquito larvae photos and then register for this webinar. And I'm going to stop sharing, and I have the other questions for you, Ryan.
Okay.
How are your population numbers with CDC transparent based on the HIPAA statute and COVID?
Yes, so none of the human cases that were shown were mapped. So, we have a whole IRB protocol as far as that. And so even data, when we publish it in a manuscript, it has to go through all those sort of guidelines and things like that. So none of the stuff that, I showed here, you could trace back to a specific household or things like that. But as far as simply taking a photo of a mosquito, whether it's larva or adult, there's no sort of HIPAA concerns until you get into the realm of human case data, which is a whole different ballgame. And that's usually the biggest bottleneck in any of these sort of studies for, disease modeling. For example, the West Nile virus for DYCAST in California, I happen to be working at the California Department of Public Health. And so, had greater access, although I still had to go through, all those steps to get the human case data. But you do need that case data if you're running DYCAST on something like dengue or ZIKA. And that's one of the reasons that we wanted to develop this tool so that you can download it and run it yourself on your own computer. Or if you work for a health department, it can be, locally on your own computer running data that only you have access to, but it's with free, open source software on QGIS as a plugin. And you'll be able to do all of that yourself as opposed to, sending it off to us or someone else. Sending all that human data just doesn't really tend to work. It's really hard to get those sort of permissions. So that's exactly one of the reasons that we wanted to develop this, so that if you do have access to the cases yourself, you can do this sort of analysis with the free and open source software.
Okay. For AR and GIS, are you using ArcGIS and NASA models for geospatial satellite imagery with your GitHub repositories?
So they've been kind of separate. So the GitHub stuff is all DYCAST related. So if you go to the DYCAST.org, you can see the papers, some of the animations, and links to the GitHub. All the remote sensing imagery, so the stuff that I showed from Connor's thesis, that was all the aerial photography, that 0.5 meter super high resolution, that was flown from aircraft. We did have satellite imagery as well. In fact, we even used the same stuff that we used to find the abandoned tires and things like that, that we just we actually purchased from a third party for that study. But there's plenty of just wonderful satellite data sets online. And Rusty, what was the one that you just told me about that was launched last summer? That wonderful satellite repository online?
Oh yeah, the
Appears,
which is an access point for a lot of the NASA data. You can also, the Mapbox also provides free access to Maxar high resolution data, which is at 5 meter scale, which is really enough to resolve a lot of mosquito habitats. Not tires, but pretty close. Yeah.
Yeah. Well, that's great. Yeah, I haven't used the Appears data yet, but we definitely will be.
Yeah.
That'll be on Connor's to-do list this summer.
Yay.
I think that wraps up most of the questions that I've got here that I've seen. Very enthusiastic support for your presentation and excitement all around from everyone. So thank you very much, Ryan.
Thank you. Thanks for having me.
Thank you also very much, Amelia, for your time, and we're looking forward to your presentation next month. Oh, Lisa has one last question. Are you taking into consideration any other vectors such as no-see-ums or horseflies?
No, possibly down the road. I mean, with that anatomically informed deep learning, we should be able to detect non-mosquito species like the bee results that I showed. So it's possible. It's just a matter of time and effort. So right now for this project, we're primarily focused on mosquitoes and those particular invasive species and vectors, but a lot of this technology would have broader application to other vectors and other insects as well.
Great. Well, thank you all very much for your time. And the webinar recording will be edited and posted up on our GLOBE Mission Mosquito website shortly. So if you missed it or have further questions about it, you can replay and watch it. So thank you very much to all of our presenters and our hosts and our participants for attending. It was great.
Thanks so much, Ryan.
Thanks for having me. Take care, everybody.
Take care. Thank you. Bye-bye.